Very quickly, Heather, do we dare attempt a polling question, or will things break down? We're going to, oh, it looks like there's already been some response to the polling question.
People have been seeing it answering, so we have some feedback.
That's fantastic. Let's look at polling question number 2, if we can get to that slide. We asked which of the following best describes your organization's current level of automation. So assuming that unbeknownst to us, the question has been answered, let's take a look at the results. It looks like islands of automation and some manual processes is the lead here. David and Irina, I'm just going to ask you to look at these responses to this question and tell me quickly if you're surprised or if you have any commentary on these responses.
I'm not surprised that we still see a lot of manual and islands of automation, because there was a Biopharma, I believe it was a survey like a couple of years ago where they found or concluded that 80% of the companies are actually at that stage islands of automation or even manual. So I'm not surprised.
Yeah.
And that goes back to some resistance of bringing technology to, for example, have automated batch records. You know, things that look simpler on an R&D level, that you can have electronic notebooks. When you go to the GMP space, everything becomes a lot more complex and, of course, regulatory driven. And there's concerns about, are we really ready for those implementation? Islands of automation also, because it's expensive to have a full distributed control system that incorporates every single equipment you have in your facility. So that goes back to millions of dollars of investment that facilities or even small companies are now willing to have, because in a couple of years, a particular piece of equipment becomes obsolete and they want to replace it. So now you have to add a lot more investment to bring that new piece of equipment, the new version of software into the distributed control system. You see, so you have this puzzle and you have this vision that looks really nice, but requires a lot of investment on a digitalization. So the question we should ask ourselves across the industry, includes vendors and small and large, is how can we have these equipment a little bit more blind, developed in a way that is like blind to the way they integrate both with distributed control systems and with themselves within the facility and with the next generation. So we're not just depending on new patches of software that are extremely expensive.
Yeah, and often companies underestimate the effort needed to get to a fully automized environment. It takes a lot more energy and time and resources, and also expertise, because all of a sudden you're not just working with bioprocessing experts, you have data science and a lot more people. So it can get very quickly complex.
Yeah. I want to get to a question from Richard. Richard, you've been following along diligently. I don't know if you guys have noticed, but he's answered true or false to some of the questions that I've asked. Richard, just so you know, I've got your number, and we're definitely going to invite you to be a panelist on our upcoming event. But let's take a couple of his questions. I like this one. What are the control strategies to align upstream and downstream for product and process related impurity monitoring to ensure batch release? Irina, can you grab that one before you have to head out?
That's a good one, Richard. I'm not going to say it's $1 million question anymore. I think there's some answers that we should absolutely refurbish the control strategies that you already are using on your batch process. But by rethinking them, you don't want to make them more complex. You want to streamline them across unit operations. So think about when the fed, this is a very simple way or exercise for a strategy perspective. Your fed batch is operating, I'm going to assume, for 2 weeks. If you were to pull the material for the first 5 days, it would be different than when you pull the material at the end of your 2-week production. And then you're going to go ahead and you harvest, and now you have a pool of material that is homogeneous because you had the opportunity to pull it together. We all agree that when a perfusion bioreactor is also running, and until it reaches a steady state, and some of them don't even reach a steady state, they are dynamic, they're changing, and they're still acceptable. If you were to pull that entire material at the end of the production, you would also make an homogeneous pool. Because you're not really purifying the pool like you do for fed batch, now you have to think about, is my downstream process enabling the purification of product and process-related impurities across the duration of the perfusion in a way that when you pull the material at the end, at the entire end of purification, it's going to be homogeneous and it's going to meet your specification. The problem comes, and I'll pass it to David, if there is a disruption due to either you divert some of the material due to contamination, or something breaks, or a cycle in a purification column goes wrong, right? And you take some of that fraction of material, you need to assess now the quality of your product without those few days that you had to remove the material from the full pool. And that's where your strategy for control needs to focus on.
Anything to add there, David?
Yeah, I think the fundamental difference between, and you highlighted, Irina, between a fed batch process and the continuous is that in the fed batch process, your product or your product quality may change or is changing because the conditions change between day 5 and the end of the culture. Whereas in the continuous mode, you operate in what we call a semi-steady state, and you have a much more controlled way of running your culture, and very likely, if you do it well, your product quality is much more stable at the end of the day. And this will definitely then change the way you run your downstream train and the way you control whether you run a fully continuous downstream or it's still a batch, but it definitely will then change how you run your downstream.
Very good. Yeah. Dr. Ramos, are you still with us?
Yep.
Oh, okay.
Yes.
Yeah, I know you need to leave. Do you want one more question, or do you need to jump off?
Quick one, maybe, and then I'll adjourn, and thank you again for the opportunity.
Oh, I appreciate the opportunity. I'm gonna hit you with one of the true or false questions so you can sign off with this. True or false, fed batch systems are more competitive at smaller scales, like 100 kilograms a year?
You know, that's so hard to say true or false. What is competitive in our market? I think it goes back to, can you be productive in continuous? Because that 50 kilogram can be a very small footprint in your facility if you run continuous. So while I want to answer your question, I also want to put a caveat that think about productivity in the bioreactor before the complexity of the process, and then add the complexity of the process. If you think about the success of your biology within the perfusion mode of operation, that shrinks everything else and makes everything much easier to control, you're going to ultimately need less people and leveraging automation in a simplified way. So yeah, I'm not answering your question, but hopefully that gives food for thought for others not to be just stuck with the simplicity of one versus other and cluster that into scale.
Yeah, we acknowledged from the outset that there's a lot of nuance and gray area here, so there's no expectation that we're binary in our true-false responses. And Dr. Ramos, I know that you're doing important work on the road right now, and you know how much I appreciate you. I've told you many times. You know how much I appreciate you, but I just want our audience to know that I appreciate your fortitude in joining us today and making this work. Thank you so much, and we will let you get back to the important work you're doing. And thanks again for joining us.
Have a good day. Yeah, thank you.
Yes, you too. Thanks. If I may add something to this, to Irina, because it goes well with Richard's next question: is it worthy to develop continuous manufacturing for products to treat rare diseases or gene therapy? I think this is a really good question, because Irina said it well. It depends. If you have only 50 kilograms per year, it's a very small footprint. So the question is, does it make sense to add more complexity, or is a fed batch process just good enough? And to give you some numbers, there's a good UCL study, the one by Susan Ferret and her team, they noticed that above 100 kilograms per year to up to 500 kilograms, there is a definite advantage of continuous manufacturing, but below, not so clear. Again, I would say depends on your molecule, on your productivity. Is it also a rare disease or not? And then, yeah, between 100 and 500 kicks, definitely there it makes sense. You have cost savings of at least 30%, and then above it goes down again because oftentimes you have to run several parallel trains. And then it can go down to 10%, but still, even at larger volume, there's definitely an advantage of running continuous.